Hey everyone, how's it going? I'm going to talk to you about the next phylum that we need to study here in our zoology lab, and that's the Nematoda, or the nematodes. And so, just to put this in context, you'll recall that the phylum Platyhelminthes had the flatworms, and then the phylum Annelida had the segmented worms, and so now the phylum Nematoda has the roundworms, okay? And this is a very important group, specifically because of disease. Many of these are responsible for diseases in humans and in livestock and in plants. So the nematodes, the roundworms, are something that we really need to understand from a public health perception. Now, Nematoda sounds a lot like Trematoda. Make sure you get these straight in your brain. Trematoda was a class within the Platyhelminthes, right? Trematodes are a type of flatworm, the flukes, which also are parasites of vertebrates and also can cause disease, but you got to keep these things straight in your head. And you'll recall that we called all of these things worms. We tend to think, anything long and skinny, we call it a worm, but that doesn't mean that they're all closely related. Another term that's an older term that you're still going to run into, the parasitic worms like the flukes, the tapeworms, the roundworms, they're often called helminths. And medicines that are used to kill them are often called anti-helminthic or something like that. And I think that's probably left over from, they used to maybe be all classified together because they all were worms, but now we know that these are not all necessarily closely related to one another. Now, if we look at our phylogenetic tree, we can see this, right? And so when we talked about the annelids, we said that, they're closely related to the mollusks, and you wouldn't really picture that. But now these roundworms, these nematodes, these are a worm-like creatures, but they're not very closely related to the annelids, all right? So if you look, they have a more distant common ancestor. And so even though they're worms and they have some similarities, they're not super closely related to one another. Now, the nematodes are the first in this group here that includes the arthropods, and this group has an exoskeleton that is shed. And we have not seen that yet. So that's a new characteristic, and that's why this clade is sort of set off from the earlier clades, because we've got this new derived characteristic. And so we'll see that in some of our next groups. Certainly, we'll see it in this group and in the arthropods. Okay, so that exoskeleton is also called a cuticle. It's non-living. It's not made of cells, but it is excreted by the cells. And it's usually made of something like chitin a lot of times. And so because it's a hard exoskeleton, it must be shed or molted as the organism grows, right? So you can't get bigger until you get rid of that small exoskeleton. This allows the roundworms to have a very rigid hydrostatic skeleton. So we talked about the hydrostatic skeleton in the annelids last week and how that is used to sort of help them move. Now, because we've got this hard cuticle on the outside that allows the roundworms to have a very rigid hydrostatic skeleton. But we'll also contrast their movement with the movement of the annelids. In the annelids, we said you had circular muscles and longitudinal muscles. And so you remember how the earthworm would move. It would squeeze and elongate a segment, and then it would relax, and that would pull the worm forward, but then it could also turn left and right. These roundworms don't have those circular muscles. They just have the longitudinal muscles. And so they can just kind of move left and right, and they have a very stereotypic whip-like fashion. And so, I'll show you some videos of these in lab, and when you see them, they've got this very characteristic movement. And so here's a figure from your book, just kind of showing some of that structure. And you'll notice down here, let me get my pen. But you'll notice down here, you've got these longitudinal muscles that when they contract, they can make it whip left and right, but they don't have any circular muscles. Okay, so let's talk about some of these roundworms and the diseases that they can cause in humans. Ascaris lumbricoides is a very common, well-known roundworm. It's called the large human roundworm. Maybe over 1 billion people in the world have this, so it's a very common problem. The eggs live for a long time in the soil, and humans tend to get this by ingesting soil or unwashed vegetables. And then once the eggs get inside, they hatch. The juveniles will burrow through your intestines to get into your bloodstream and your lymph vessels. And from there, they'll move to the heart and to the lungs. Then when they get into the lungs, they'll break out and move into the trachea. And then they get kind of coughed up, or they'll crawl up, they'll get into the esophagus, and they'll get swallowed a second time. This time they'll be swallowed as juveniles. The first time it was the egg that was swallowed, then it migrated, through the body, gets coughed up, gets swallowed as a juvenile. Then when in the intestine, it will grow into an adult, and the adults will reproduce and then shed eggs and complete the cycle. But those adults can get very thick in the intestines and get so thick that it can actually block the intestines. And the adults might even actually crawl out, and that might be a symptom or a sign that you've got these roundworms. Anyway, this can cause a severe blockage, which can be a problem. And here's a figure from your book showing the swine version of this. And so this is a swine intestine that's cut open. And you can see the huge number of these roundworms that would block this intestine. Another type of roundworm that is found often in dogs, Toxocara canis. And so, many times you give your dog a, wormer, or you have to deworm your dogs. And sometimes it's for heartworms, which we're going to talk about in a second here. But there's also just these intestinal roundworms, or different kinds of roundworms that you can also, the dogs can also catch. And so these are unique in that a lot of times the adult dogs are carrying them, but because the adults have a well-developed immune system, the roundworms don't really develop very well. So the adult is carrying it, but it's not a full-blown infection. However, if you've got a female dog that has these and she gets pregnant, then those changes often stimulate the roundworm to then start developing more. And so then the puppies can be born infected with this roundworm. And this is a very common way to transfer this nematode to humans. And it's often through children, right? You play with a puppy, or you're around feces that are contaminated with the roundworm eggs or whatever. And so this is a common vector of transmission from dogs to humans. And so even if you didn't even know that the mother had the worms, but the puppies are born with the worms. One of the things that this can cause is called ocular toxocariasis in humans. And basically it's when the worm crawls into the back of the eye. And so you can get blurry vision and floaters. And so here's a picture of the back of someone's eye, and it shows what's known as a granuloma. And so a granuloma, you can see this kind of whitish streak on the back part of the eye, but you can see how that's shaped like a roundworm. That's where that roundworm was burrowing and sitting in the back layer, in the layers of skin in the back of the eye. And so, of course, that can be a problem if you've got these worms in your eye, and that's one of the symptoms of this, or one of the characteristics of this disease. Hookworms are another type of nematode that's a very common, humans are very commonly infected with. Their life cycle is very similar to the intestinal worms that we talked about earlier, but instead of swallowing the eggs, humans tend to get infected by the worms burrowing through their skin. And so it's not a matter of hygiene or not having access to clean water. Just walking around barefoot, you can pick up these hookworms. And so here's a figure from your book kind of showing you how the mouth of one of these hookworms, and this is how they're able to burrow through your skin. They've got these sharp plates that they can dig through. Now, hookworms used to be a big problem in the U.S., but improvements in hygiene have really reduced the numbers, specifically with sewage treatment. And so many people aren't exposed. The eggs aren't around very much, which is why it's not, anymore in the U.S., it's not a big deal to walk around barefoot. Don't worry about getting hookworms if you're walking around barefoot outside. Here's a figure from your book showing the life cycle of these hookworms, and you can see that they're showing that the juveniles here burrowing through the skin to infect a human. And then, just like before, once they get in and get into the bloodstream, then they move to the lungs, and then they break out of the lungs, which they're showing here, moving from the blood vessels into the alveoli of the lungs. They get coughed up, and then get swallowed and get into the intestine again. And once in the intestine, the juveniles develop into adults, and then the adults mate and they lay eggs. And so again, if the eggs are shed in the feces, and so if you have untreated sewage or septic tank runoff, that may be a way to get exposed to them. Or in some cultures they use human feces to fertilize fields and that's a way to be exposed to the eggs, or be exposed to the hookworms in the ground that can crawl in and burrow through your feet. But like I said, here in the United States, we don't do a lot of that. We've got good sewage treatment. We don't use human feces as fertilizer, and so we're not really exposed to these. So again, don't be afraid to walk around barefoot. Now, these hookworms play a part in this interesting idea that's floating around, the hygiene hypothesis. And so this is something we talk about every once in a while. It's an interesting idea. And basically this says that as a sanitation improves, our immune systems don't get challenged like they used to. Our immune system evolved in a very different environment than the one in which we live now, right? There are a lot more microbes and a lot more challenges to the immune system. But here in the United States and in other more developed countries where we have better sanitation, then we don't have as many challenges to our immune system. But when we do get challenged, our immune system overreacts. And that is what an allergy is. And so that's the hypothesis is that's why we see more allergies in more developed countries, because an allergy is an overreaction of the immune system. And so the hygiene hypothesis suggests that the immune system needs to be, quote unquote, calibrated so that it responds when you have a pathogen, something that it should kill, but it doesn't over-respond, and it doesn't respond to symbiotic microbes, because you have a lot of things that live in your gut that are very important for you, and you don't want your immune system to attack them. Now, this is, very much an oversimplification, but you get the idea. And so the relationship to hookworms is that if you purposely infect someone with hookworms, they won't get very sick from the hookworms, but your immune system's reaction to the hookworm and also the hookworm's reaction to your immune system helps to moderate your immune system reactions. And so by infecting people with these hookworms, you actually might make it so that they have fewer of these allergic reactions. And so this is an interesting idea. The thing is, it's been tested, but not successfully. So it's a good idea, but it's never really been shown. There's no data to back this up. One counter argument to the hygiene hypothesis is that lots of people are infected with roundworms or with hookworms, and they still have allergies, right? And so you would expect those people to have fewer allergies, and we don't see that. There's lots of claims about this working, and but there's not very much data that suggests that it works. So it is an interesting idea. It is one of those things that helps us to understand our immune system response more fully, but there needs to be a lot more study on this, and certainly there needs to be a lot more study to ensure the safety and effectiveness of something like this. But it is an interesting idea. Okay, another nematode that you may have heard of is Trichinella spiralis, which causes the disease trichinosis. Trichinosis is something that humans often get from eating undercooked pork. And so if you've got a swine that's infected with this roundworm, the juveniles are in cysts in the muscle. And if you don't cook the muscle enough, the cysts aren't destroyed. And so then when you eat the cyst, your stomach acid breaks that outer cyst covering, which releases the larvae. They develop into adults and then mate in your intestine. And then the pregnant females are what's known as ovoviparous. We're going to talk about this in class here in a couple days. That means that they form eggs, but they retain the eggs in their body. And so they give birth to live young, but they don't nourish those young from a placenta. They just have an egg that they retain in their body. At any rate, the female releases these live larvae, and then the larvae burrow into the human's muscle and then encyst, just like it did in the swine muscle. And so that is trichinosis. So that's just caused by another species of nematode. Very common roundworm infection are pinworms, and these are found in lots of humans. Again, this is an intestinal parasite, and when the female is ready to lay her eggs, she migrates to the anus and then lays eggs right around the anus, and this causes an itch and an irritation and causes the human to scratch that itch, and that's how the eggs get spread around. By scratching, the eggs get shed, and then by sharing a bed with an infected person or not cleaning your sheets, that's how these eggs can spread from one person to another. Again, this can be a fairly common human parasite. The last group we're going to talk about, the filarial worms. And these are roundworms that use mosquito as a vector of transfer. And so we've talked about different ways that humans can get infected with these different kinds of nematodes. These move from a mosquito, an infected mosquito will transfer them to a human or another mammal, and then when an uninfected mosquito bites that person, they can pick them up, and that's part of the life cycle. A common disease that's associated with these types of worms is elephantitis. There's a couple different roundworms that might cause this. And so here's a figure from your book, again, sort of showing the life cycle, and you can see the mosquito getting infected, and then passing the infection on to another human. And then once those larvae get into the bloodstream, they migrate to the lymph nodes, and they develop into adults. And the adults block the afferent vessels to the lymph nodes. And so let's talk about this. What is lymph? What are lymph nodes? Lymph is part of your immune system. And so it's a clear liquid that's got lots of white blood cells. And the lymph nodes are areas where this lymph gets filtered. And so it's part of your immune system response, and it helps to catch and filter out pathogens that may be infecting your body, right? But in this instance, you've got the pathogen or the roundworm that's actually blocking the afferent vessels or the incoming vessels to these lymph nodes. And so the lymph can't properly drain, and that causes elephantitis. So this is some terminology that we need to know, because this is going to come up in other areas. Afferent vessels carry lymph to the lymph nodes, and then efferent, which is not labeled here, but this is the efferent vessel to the lymph nodes, that carries lymph away from the lymph node. These terms are used in other areas, for example, neurons, and so afferent usually means carrying something to, and efferent usually means carrying something away. Couple ways to try to remember this that may help or may not help. Afferent approach, while efferent exit. So think about this lymph node. The afferent things are approaching the lymph node, and the efferent is where they exit. A always comes before E, and so if whether it's a signal on a neuron or whether it's lymph nodes, the afferent always comes before the efferent, or also the efferent equals an effect. And so when we're talking about neurons, the efferent neuron usually causes the effect. It causes the response. So these are ways that might help you remember the difference between afferent and efferent. In this particular example, you see the afferent vessels are plugged up, and so the lymph can't properly drain, and so you get this very painful swelling that is sometimes called elephantitis. Now there's another so there's another type of roundworm, and I don't think this is a filarial worm, but it's also an important human disease. It's something called guinea worm disease. And so the larvae of this, again, this is not that same filarial as the last one that causes elephantitis. These larvae live in organisms called copepods. Copepods are small crustaceans. They're arthropods. We're going to talk about them in the next phylum. But these live in water, and they get infected by this roundworm. And then if a human ingests the water with these, that's contaminated with these infected copepods, the larvae move into the humans, develop into adults, reproduce inside the human, and then the female migrates to the skin in order to get rid of her eggs. And so when she gets to the skin, she breaks through the skin, and it causes this very painful blister. And if that blister is submerged in water, then the female releases her larvae into the water, and that helps to complete the cycle. But this blister is very painful, and that's usually the point where the person realizes that they've got this guinea worm disease. And so here's what it looks like. When that worm is at this blister, that's when you have to get hold of it and try and pull that worm out. The problem is you have to be careful, because if you break the worm, the pieces that are left inside you will rot, they will get infected, and you've got a problem. So you need to be able to pull this worm out without breaking it. And so a common treatment is to wrap it around a matchstick or a stick. And so you pull a little out, you wrap it around a stick, and then each day you pull a little more out and you wrap it around the stick. So you sort of gently pull this worm out until it comes out, which is, that's pretty wicked. But it's pretty bad disease. Luckily, it's not something that's very common here in the U.S. Okay, so I'm jumping back now. Another type of filarial worm are dog heartworms. So again, remember, these filarial worms are roundworms that have a mosquito as part of their life cycle. But dog heartworms are, a common disease in our pets and our canines. You may be familiar with them, and they are what they sound like. They are nematodes that migrate to the heart. And here's a picture, here's another picture, and of course they can get very thick and cause severe blockages, and these can be a really dangerous health problem for your puppy dogs. Because of that, many of us have medicine that we give our dogs to prevent heartworms. And so I'd like to take this opportunity to kind of talk about one of these common heartworm preventatives, because it starts to give us a good insight. It starts to tie things together that we've been learning all semester. It's very interesting. And so these preventatives, first off, only work if your dog is heartworm free. They work at the larval stage. If your dog already has adult heartworms, then there's a very serious, very specific treatment that must go through to get rid of those heartworms. But this also is why before you start your dog on these preventative heartworms, they test to make sure that they don't have the heartworms, because this only works if the dog starts out uninfected, right? And so these preventative medicines target the larval stage. A very common one is ivermectin, all right? And so ivermectin works by binding to gated chloride channels of the muscle and nerve cells of invertebrates. So let's, think about each term in this and break them down, right? We're talking about invertebrates. And so, of course, your nematodes are invertebrates. And this ivermectin works on other phyla as well, all right? We're looking at the muscle and nerve cells of these invertebrates. And so, of course, Those are very important cells to the invertebrate parasite. And it is a medicine that comes and binds to these gated chloride channels, all right? And so when that medicine binds to these channels, that holds the channels open. So they're gated, which means that they can sometimes be closed, sometimes be open. The medicine means they're open all the time. And these are chloride channels, and so they allow chloride to flow into the muscle cell or into the nerve cell, and we call that hyperpolarization. And so this hyperpolarization means that the muscles or the nerves can't function properly. And so this causes paralysis, starvation, and ultimately death in the invertebrate. Now, mammals don't have these type of chloride cells, this particular type of gated chloride cell, and so that's why this is fairly non-toxic to humans. And so this is a medicine that's used like in shampoo to help cure lice. It's something that you can take or your doggy can take, and they don't get sick because we don't have these particular types of chloride cells. Now let's talk about this in a little bit more detail, because again, this brings together many topics we've already discussed, and it also will segue us into some of our upcoming topics in lecture. So here's a schematic of the cell membrane of a muscle cell or a nerve cell, all right? And so that's what we see here. This is the membrane, and that separates the outside of the cell from the inside of the cell, right? That's what membranes do. Now look at what I've drawn here. I've got charged ions, and you'll see I've got a lot more positive ions outside the cell and a lot more negative ions inside the cell, right? And so we say that that membrane is polarized, meaning it's got a difference in charge on either side of the membrane. And so how does this happen? How do we get more positive outside the cell and more negative inside the cell? Well, think back to lecture 9. We talked about sodium-potassium pumps. And these sodium-potassium pumps use ATP to pump 2 potassiums in to the cell, but for every 2 potassiums that come in the cell, 3 sodiums go out the cell. And so as these sodium-potassium pumps run, your net effect is you're getting more and more positive ions outside the cell. That's how we get this polarized membrane. And so the end result is that at rest, the inside of the cell is about -70 millivolts relative to the outside, all right? So volts is a measure of difference of potential. And since the inside of the cell has more negative, it's about -70 millivolts inside the cell relative to outside the cell. And this is very important for nerve and muscle cells because they must be polarized in order to function properly. This is something we're going to talk about in lecture. How, why do they need to be polarized? How do they function? We're going to get to that. But just trust me that they have to be polarized in this manner to work. Now, although there's, a net positive charge outside of the cell, there's lots of negative chloride ions outside the cell, but there's not much chloride ions inside the cell. And so, in general, there's a lot more, sodiums, and there's a lot more potassiums, or a lot more sodiums outside. And so you've got a net positive outside the cell, but mixed in here, you see a few negatives. You've got these chloride ions, which have a negative charge. But inside the cell, there aren't many of these chloride ions. So what you have is you have a high concentration of chloride outside the cell and a low concentration of chloride inside the cell. So what have you got? You've got a concentration gradient. And so what happens when you have a concentration gradient? You get diffusion, right? Remember we talked about diffusion. So the chloride wants to diffuse from outside the cell to inside the cell because it wants to travel down its concentration gradient. But it can't because you've got this semi-permeable membrane that won't allow it to cross. But it can cross if it has the proper channel. And so that's where these gated chloride channels come in. These gated chloride channels come in, are used to allow chloride to come into the cell. When the gate's open, the chloride flows in. When the gate's closed, the chloride can't flow in, right? And so again, go back to lecture 9, we talked about gated channels, right? Proteins that are embedded in the membrane that can either be open or closed. Okay. And so in this invertebrate, in this heartworm, or in this louse, or in any kind of invertebrate parasite, you've got those gated chloride channels in the membrane of the nerve and muscle cells. And that heartworm preventative goes and binds to that gated chloride channel and holds it open. All right? And so now, all along this membrane, all those gates are open. Well, what's going to happen? If the gates are held open and you've got a difference in concentration in chloride, chloride is going to flow into the cell. There's nothing to stop it. It wants to diffuse in. There's nothing to stop it from diffusing in. So now we've got a lot more of that chloride ion inside the cell than we originally had. What's that going to do to the charge inside the cell? Well, of course, the charge is going to become more negative because you're adding a lot more negative ions to the inside of the cell. So although originally we were at like negative 70 millivolts relative to the outside, now we're at, say, something like negative 80 millivolts, right? Which we're more negative inside the cell. That is what we call hyperpolarization. The negative 70 was a polarized membrane. Now we are hyperpolarized. We're even more negative. There's even a greater difference in potential between the inside and the outside. And so that ultimately is how this medicine works. So again, those nerve and muscle cells of the parasite must be polarized to function. This is true of any nerve and muscle cell, right? All nerve and muscle cells need to be polarized. And so the inside needs to be about negative 70 millivolts relative to the outside. How do we get this polarization? Sodium-potassium pumps. At the same time, outside the cell, you've got a lot of chloride ions, which are negatively charged. But there's a lot more of this sodium outside the cell. So that's why the outside of the cell, even though there's lots of chloride, it's still positive outside the cell because there's a lot of sodium outside the cell. Now that heartworm preventative binds to the chloride gates and it holds those gates open. Now it only does this on the nerve and muscle cells of the parasite. So when those gates are held open, that allows that chloride to come in. That makes the cell hyperpolarized, right? And again, we don't have this type of chloride gate. We have chloride gates in our nerve cells and our muscle cells. We just don't have the same kind. You know, it's a different protein. And so this heartworm preventative will bind to the chloride gates of the invertebrate, but not of the vertebrate. And that's why it's relatively, it's presumed to be non-toxic for us. So now that we have this hyperpolarized nerve and this hyperpolarized muscle, they can't function properly. And if they can't function properly, then the larvae, whether it's a roundworm or a louse or whatever, is paralyzed, starves, and dies. And so that's, a little bit of a long story, but I wanted to tell you that story on purpose because, like I said, it starts to pull together a lot of the things that we've been learning. And now you're starting to see a method to the madness. Why did we learn about things like the membrane? And why do we learn about gated channels? And why do we learn about diffusion? Why do we learn about proteins and their shape? Well, now we're starting to pull it all together. And an easy way to pull it all together is to talk about things like medicine and disease, and you can start to understand how these things work. So anyway, I think it's very interesting. I think the nematodes are interesting. We've seen several examples. We're going to look at some in lab. That's all I have for right now. Thanks for listening, and I'll see you later.